Hello dear students. Today we'll discuss about the chapter Heredity and Evolution, and in this chapter we'll be discussing about just the part heredity. So let's start now. We have seen that reproductive processes give rise to new individuals that are similar but subtly different. In this chapter, we shall be studying the mechanism by which variations are created and inherited. The long term consequences of the accumulation of variations are also an interesting point to be considered. We shall be studying this under evolution. Inheritance from the previous generation provides both a common basic body design and subtle changes in it for the next generation. Now, in this figure, it represents the situation if a single individual reproduces, as happens in asexual reproduction. If one bacterium divides and then the resultant two bacteria divide again, the four individual bacteria generated would be very similar. There would be only very minor differences between them generated due to small inaccuracies in DNA copying. However, if sexual reproduction is involved, even greater diversity will be generated, as we will see when we discuss the rules of inheritance. The most obvious outcome of the reproductive processes still remains the generation of individuals of similar design. The rules of heredity determine the process by which traits and characteristics are reliably inherited. Now, let us take a closer look at these rules. But the question is, what exactly do we mean by similarities and differences? We know that a child bears all the basic features of a human being. However, it does not look exactly like its parents, and human populations show a great deal of variations. Let us take an example to understand this better. You all must have observed the ears of your friends and your classmates and your near and dear ones. Now, prepare a list of those having free or attached earlobes and calculate the percentage of students having each. Now, find out about the earlobes of the parents of each of your friends who are all in the class. Now, correlate the earlobe type of each of your friend with that of their parents. Now, based on this evidence, you suggest a possible rule for the inheritance of earlobe types. Now, the number of people having the attached or detached earlobe, which has the maximum number, we'll say that that is the dominant trait in that particular population. Now, let's look at another example. For example, of rolling and unrolling of the tongue. There are some who can roll their tongue and some who cannot. So, you can try whether you can roll your tongue and can or you cannot roll your tongue and see whether your parents can do the same or not. So, this trait as well is inherited from the parents. The rules for inheritance of such traits in human beings are related to the fact that both the father and the mother contribute practically equal amounts of genetic material to the child. This means that each trait can be influenced by both paternal and maternal DNA. Thus, for each trait, there will be two versions in each child. What will then the trait C in the child be? Now, Mendel worked out the main rules of such inheritance, and it is interesting to look at some of the experiments from more than a century ago. Mendel was educated in a monastery and went on to study science and mathematics in the University of Vienna. Failure in the examinations for teaching certificate did not suppress his zeal of scientific quest. He went back to his monastery and started growing peas. Many others had studied the inheritance of traits in peas and other organisms earlier, but Mendel blended his knowledge of science and mathematics and was the first one to keep count of individuals exhibiting a particular trait in each generation. This helped him to arrive at the laws of inheritance. Mendel used a number of contrasting visible characters of garden peas like round and wrinkled seeds, tall and short plants, white and violet flowers, and so on. He took pea plants with different characteristics, a tall plant and a short plant, produced progeny by crossing them, and calculated the percentages of tall or short progeny. In the first place, there was no halfway characteristics in this first generation or F1 progeny, so no medium height plants. All plants were tall. This meant that only one of the parental traits was seen, not some mixtures of the two. So the next question was, Were the tall plants in the F1 generation exactly the same as the tall plants of the parent generation? Mendelian experiments test this by getting both the parental plants and these F1 tall plants to reproduce by self pollination. The progeny of the parental plants are of course all tall. However, the second generation or F2 progeny of the F1 tall plants are not all tall. Instead, one quarter of them are short. This indicates that both the tallness and shortness traits were inherited in the F1 plants, but only the tallness trait was expressed. This led Mendel to propose that two copies of factor, now called genes, controlling traits are present in sexually reproducing organisms. These two may be identical or may be different depending on the parentage. A pattern of inheritance can be worked out with this assumption, as shown in this chart. Now, when you look at this chart, we can see that the F1 progeny is being self pollinated. In this chart, you can see the male uh, progeny and the female progeny is being crossed here, and the cross of between these progeny produces three tall plants and one short plant in the ratio. This is the phenotypic ratio, which shows the physical appearance that physically these are tall uh, and one is short. But the genotypic ratio is 1 is to 2 is to 1, which tells that 1 is the pure tall, 2 are the impure tall, or we can say they have mixed genes in which one is the dominant and the other one is the pure short plant. In this, both capital T and capital T and small t are tall plants, while only small tt is a short plant. In other words, a single copy of capital T is enough to make the plant tall, while both copies have to be small t for the plant to be short. Traits like capital T are called dominant traits, while those that behave like small t are called recessive traits. Now, what happens when pea plants showing two different characteristics rather than just one are bred with each other? What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? They are all tall and have round seeds. Tallness and round seeds are thus dominant traits. But what happens when these F1 progeny are used to generate F2 progeny by self pollination? A Mendelian experiment will find that F2 progeny are tall plants with round seeds, and some were short plants with wrinkled seeds. However, there would also be some F2 progeny that showed new combinations. Some of them would be tall but have wrinkled seeds, while others would be short but have round seeds. You can see as to how new combination of traits are formed in F2 offspring when effectors controlling for seed shape and seed color recombine to form zygote leading to form F2 offsprings. Thus the tall or short trait and round or seed or wrinkle seed traits are independently inherited. In this experiment, we can see the combination or breeding between the uh, plant having round and green seed with wrinkled and yellow seed. And in the F1 progeny, all the seeds produced are with yellow and round seed. That means the yellow color of the seed is the dominant and roundness of the seed is also the dominant trait. And when the F1 progeny is self-bred, so we get a mixture of these traits as we can see in the chart. Here we can see that the breed with dominant traits are maximum in number and the breed with recessive trait only is very less in number. And the breed with mix, that is dominant as well as the recessive trait, are in average, right? Like the seeds with the round and green seeds and wrinkled with yellow seeds are 3 is to 3 in number, whereas with all yellow and round seeds are in 9 ratio and the one with the wrinkled and green seed are in the 1 ratio. So the ratio is 9 is to 3 is to 3 is to 1. So the three laws that were framed by Mendel by after doing these experiments, that is after doing the monohybrid cross that is taking just one trait and the dihybrid cross that is taking two traits at a time, they were law of dominance, law of segregation, and law of independent assortment. So the law of dominance says that a dominant allele completely masks the effects of a recessive allele. A dominant allele produces the same phenotype in the heterozygotes and in the homozygotes. The law of segregation says that in a diploid organism, chromosome pair are separated into individual gametes to transmit genetic information to offspring. And law of independent assortment says that alleles on different chromosomes are distributed randomly to individual gametes. Now let's talk about how do these traits get expressed. Cellular DNA is the information source of making proteins in the cell. A section of DNA that provides information for one protein is called the gene for that protein. Now, how do proteins control the characteristics? Let us take the example of tallness as a characteristic. We know that plants have hormones that can trigger growth. Plant height can thus depend on the amount of particular plant hormone. Thus, the amount of plant hormone made will depend on the efficiency of the process for making it. Consider now an enzyme that is important for this process. If this enzyme works efficiently, a lot of hormone will be made and the plant will be tall. If the gene for that enzyme has an alternation that makes the enzymes less efficient, the amount of hormone will be less and the plant will be short. Thus, genes control the characteristics of trait. We have discussed the idea that the two sexes participating in sexual reproduction must be somewhat different from each other for a number of reasons. Now, how is the sex of a newborn individual determined? Different species use very different strategies for this. Some rely entirely on environmental cues. Thus, in some animals, like a few reptiles, the temperature at which fertilized eggs are kept determines the type of sex that reptile will have. Now, whether the animal developing in the eggs will be male or female. In other animals, such as snails, individuals can change sex, indicating that sex is not genetically determined. Some species even have no sex determination system. Earthworms and some snails are hermaphrodites. A few species of lizard, fish, and insect are all female and reproduce by parthenogenesis. However, in human beings, the sex of the individual is largely genetically determined. In other words, the genes inherited from our parents decide whether we will be boy or girl. But so far, we have assumed that similar gene sets are inherited from both parents. If that is the case, how can genetic inheritance determine sex? The explanation lies in the fact that all human chromosomes are not paired. Most human chromosomes have a maternal and paternal copy, and we have 22 such pairs. But one pair, called the sex chromosomes, is odd in not always being a perfect pair. But one pair of men have a mismatched pair in which one is normal sized X while the other is a short one called Y. So, women are XX while men are XY. Now, can we work out that the inheritance pattern of X and Y will be? As in this figure, it shows that half the children will be boys and half will be girls. All children will inherit an X chromosome from their mother, regardless of whether they are boys or girls. Thus, the sex of the children will be determined by what they inherited from their father. A child who inherits an X chromosome from a father will be a girl, and one who inherits a Y chromosome from him will be a boy. So, I hope you understood this chapter, and this was all in this chapter. Thank you.